Okay, just to briefly recap what we did and where we are, right? So on Monday we went sampling, take the water sample, and then on Tuesday morning we concentrated the biomass. We didn't use the pump. Do you remember what we used? Centrifuge. Yeah, we put it away with centrifuge. And then we spend a lot of time extracting DNA and cleaning it up. And so the whole of Tuesday we did DNA extraction, clean up, and then we quantified with which instrument? Qubit. Yeah. And the good news is we did have DNA. So once you have DNA, you can start working. Also, normally you have more DNA than you need. So if anything goes wrong, you can go back. So once you take the sample, you should get as quickly as possible to getting your DNA. DNA, you can put at -70 into the freezer, and 10 years later, you can go back and still use it. Yeah. And then we use the which machine for amplification? Mini PCR. Yeah. Basically, we are only interested in one gene, 16S. So we make many copies with the PCR reaction. And then this morning, we take the amplicons, we clean them up again, we prepare the library. So this is where we are. So this afternoon, we can now go into the lab and start the sequencing process. There's one step, we need to prepare the flow cell for the sequencing, and we will watch a video first. I can show you a video how this works. So just to remind you again that so what did we actually do? We have genomic DNA from the bacteria. It's a mix of hundreds of thousands of bacteria, so different pieces of DNA. And then we only want one gene in that DNA. So we have so-called primers, they can find the gene. And then we also, so F and R, that's the primer, forward and back, reverse. But we also, for each primer, we have a barcode. So this barcode tells us that this gene is from sample number 1 or 2 or 3. That allows us to put all of the samples into one flow cell. And then if we read this information, we can still know where the DNA came from. And then what's library preparation? So now at the end of each DNA piece, we need to attach another little adapter. And so basically this should help this piece of DNA strand to find the nanopore in the sequencing device. So that should now all have reacted with each other. So ideally, all of the DNA should have this adapter, should have the barcode, and the primer will still be there, and the 6-nest gene. So what are we now? What do we still have to do? Which is priming and loading the flow cell. And they recommend that we should watch this video. Okay. Okay. Yeah, so maybe we check the flow cells again. Should we watch this as well? How do I play? How can I play the sound?
Flow cells are shipped in a sealed pouch made of a moisture-proof material to better maintain the environment for the flow cell over a range of temperatures over time, as seen here on the right. The benign and gridine flow cell has 2,048 wells, and each well is designed to contain a single nanopore. Oxford Nanopore Technologies has a physical warehouse pores per flow cell of all.
Maybe should I mute mine and play there?
For
This was just. No, I know.
If this was
No, so.
QCs before using the flow cell.
yeah, okay.
Should work now. So that's did no video. Audio didn't go, I think. So you just muted now.
I locked out.
No, but you were also muted. This was also muted actually.
So you mean it was
Yeah, video was recording, but I think audio was not recording. I suppose. Program is a program where Gyan is funding lots of courses. And course is this particular course. I am course coordinator. So these are participants. I will give it to them. It's actually set. This is not, so ideally what it was supposed to be that we would have told them that we will be online this exact time, this way. So this process is different. So I mailed them just now at 2 late feeling. But then this way, I don't think anybody would get to it. Nothing more is left here, right? Okay, so I will just switch off that. So
Like this one, so maybe some of you can watch here and some can watch over here. Yeah, okay, yeah. So what it says is slide the priming port cover clockwise to open the priming port. So that's this one. I'm doing 90 degrees, just like this. So this one, just slide it 90 degrees. Yeah. See, my eyesight is actually not very good, so for that is also best. But this is the priming port. Yeah. So this thing here is the priming port. Yeah. Take care when drawing back buffer from the flow cell. So it says we should set this to 200.
Priming buffer.
So we put the 200, we put on the priming port, and then it says turn anti-clockwise, so like this. Yeah? So we put it on the priming port until it's solidly in that port. The important thing is don't press down, just turn. Yeah? Now because this is an old flow cell, maybe there's no solution coming out. So here I cannot draw any solution now. But with the new flow cell, this should be. Now the important thing is don't press like this. Okay. Right? If you press here, you press air into the flow cell. So we only turn. for now.
Yeah.
See, now some
Yeah,
came
yeah.
out. So that's enough. Yeah? Can you see here? There's a little
Yes,
bit
sir.
of liquid in
Yes,
there.
yes, yes, sir.
So that's what we want.
Okay.
Then we can remove it. Okay.
So you can use that piece.
New tip. Okay, so we did that. Okay. And then we take our priming mix. I'm just gonna take water instead. हार्दिक यहाँ पे आप there should be no air at the tip, so I don't see any,
Space
but,
नहीं होनी चाहिए टिप में और पानी में.
but we can also put like a drop
Yes.
out first, pull it into the sampling cup. And then slowly now there's some water coming out here because it's an old flow cell. That shouldn't happen in the real one. Maybe with some tissue. So we do this. But now I see some air bubbles here, huh? Oh no, it's just, I just see it. No, it's fine. But you don't want to push all the way, so there should still be some liquid in the pipette when you take it away. See, there's still a little bit of liquid in there. Yeah? So we shouldn't add any air bubbles. Okay. And then we wait for 5 to spot on 4.
Yeah, this one.
So that's this one here. So we lift it up
Okay.
like this. Yeah. Okay. So this is the sampling port, this hole here.
The transparent.
Then we do 200.
वो डालो पानी में. ये सिर्फ प्रैक्टिस कर रहे हैं. मुझे प्रैक्टिस करना है. 'यहीं से डालना है फिर वो स्पॉट ऑन से दिखेगा जाता हुआ.
Just make sure again we don't add any air in.
And we have to suck the air out. नहीं, we are not sucking it. We are putting
So
it.
we are doing the.
And we are seeing from the spot on fold. You can see there.
No, I'm saying that in every step we have to suck the air out.
No.
Yeah,
The
okay.
first step.
And you can see in this old flow cell there's liquid coming out.
Yeah.
That should not happen in the new flow cell. This was not liquid. So like this, you should start going. $200. Okay. So that's this step here. Then we take the other pipette.
अब जो लाइब्रेरी डालनी है ना वो स्पॉट ऑन से डालनी है बाकी सब टाइमिंग कोड से जाएगा हाँ
And so then 75. Why did you close this? Somebody closed this. Did it fall back? It still needs to be open. Okay. So here you would try drop by drop, just into this. Yeah, it's
Yeah.
one drop, and it should suck it in. Yeah. Now, because it's an old flow cell, maybe it doesn't suck it in nicely. And another drop. It's all off, but it should suck it in. Yeah. This anyway, and you clean it. But that's maybe because it's not the right, it's water, it's not the library. So the library has beads and so forth. So this one it sucks in. Yeah, now it's nice. Yeah, okay. So maybe you see there's like this little channel here. See these two things? Maybe that's where we should put the drops. It's empty. Okay, do you want to practice this?
yes
Yeah. Let's close it. Do it all.
हार्दिक
Please wear gloves when you do this because these old flow cells they are full of bugs. So can we get more wires like this? Together we go. Yeah, see? Now you have a little bit liquid. Yeah. So we withdraw the air from the priming core. Okay? And now you take 800 microliter of water, which again this is our
ये लो पानी ये लो एक आप भी ये रख लो जिससे वाटर ले लो कितना लेना है 800 हाँ
So you're pretty good because you don't have much water, so you could actually go almost directly.
सैलेड बबल है
Slowly, smoothly push in until almost all the liquid is inside.
It's coming from the other side.
Yeah, this is not your mistake.
You are
just because it's too clean cut this. So we might be off later.
दो तीन फोल्ड है ना समझ में
Stop. Yeah, so you stop before everything is inside. I think it's also okay.
अब स्पॉट ऑन खोल दे
So then we wait for 5 minutes and we prepare the library and then you open the spot on board. Okay? And now we need a small pipette to No, so open the stop. You need to add another 200 micrograms.
Then after that 75 now. Those are finally those are done.
Yeah, make sure there's liquid inside like you have. Then you can go to the priming probe. Timing, still the same. So you load another until it's almost all in and then you start. A little bit more.
Travel comes photo to that. That's enough, sir?
A little bit more. Stop.
Cut this please. Same पानी तो अच्छा. अब आपको ये चाहिए.
Then we take the small feedback.
75.
This is the library. And we add it drop by drop into this sample form. So I actually found these assay. Yeah, so you take, or you do it later. Just one drop, wait until it's juice. And keep at above the form. That's a drop, good. That's one drop. Just like that.
Sarah 75.
Right? Yeah, here because when we are a little bit above, we are not introducing air, so your tip should not go into the port, just drop by drop, and yes, then you can push all of it out. Yeah, this is now the let's see how it goes with your sender port. So you don't put the tip inside the thing, you just drop by drop, you add one drop after another. Yeah, like this. A little bit to the left. A little bit higher.
Yes.
Like that. Yeah. Like she does, you can use one hand to kind of stabilize your.
अच्छा कभी ये न्यू नहीं है
उसमें पहले भी सर ने भी क्या
हाँ नहीं इससे पहले भी ये यूज है
पहले भी यूज हुए
दिस इज द यूज्ड वन फॉर द प्रैक्टिस
Yeah, you're doing great. It's actually all going in. I think this cell is better than this.
Yes, sir.
Here you can see the elbows.
यूज्ड वन ओल्ड वन
They're both old. Okay, it's fine. Be ready. Perfect. Yeah. So who else? You want to try?
Yes.
Yeah, we just need to. Okay, this is the IG.
So this protects from light.
Yeah, maybe.
ये इसमें नहीं था इसके ऊपर
Yeah, so let's just do the protocol.
So that way I did it.
Yeah, if you want to do it, yeah. Oh. Press down firmly on it. I think it's fine, I guess. Yeah, now why don't you do a flow? Okay, so he's checking the flow cells, then we can prepare the buffer or the grinding mix and
So it has detected the flow cell automatically. So first it will rise the temperature to 37 degrees. This machine is basically nothing but a temperature controller. It just raises the temperature. Rest everything is done in the flow cell. So flow cell is nothing but a hydrofluidics chip. It is raising the temperature. Once this is done, then it will take 5 minutes to check all the codes. So that day we did it, it came out to be 1377 codes. So let's hope today also we try the same. Storage is always 2 to 4 degrees centigrade is enough. Always
take the 1,000 milliliter.
Everything within they have prepared the memory. They just need to first they need to add primer to some micrometers, 20 micrometers. Then they will add the library. Then we again put the machine flow cell inside and then just start it. How much time it will take? 24 hours. We can leave it for quite long, but we can stop the moment we want. Once we see that we have sufficient reads have been generated, then we can stop it. So we will let it continue to run, and tomorrow we will come and have a look how many reads have been generated. Then accordingly we will decide. Checking. This is the previous one.
Yeah, let's go to the maximum because I think this is at least 100, right? Yeah.
It is 100.
Then try and mix this, yeah? So that the beads, which are all at the bottom right now, are suspended.
We need to mix it first.
Yeah, that's what it says.
Okay.
These beads settle very quickly. It's vital they are mixed immediately before use.
So then we will be adding 25.5 microliter.
Once you've done that,
Then we will add our library.
that's good. Now we adjust it to 25.
25 and point.
Same thing. Then I would mix again a little bit, and then you take it out. 25.
Should I mix it again and then?
Yeah, I would
take
mix
that?
again, and then you take 25.
take that?
Yeah.
Okay. Okay, and now we need our library final. It's very tiny volume. So we need 12 microliters. Take a small 0 to, this is a good one.
ये वाली हो गई ना, ये टिप उसके अच्छा हाँ ठीक है.
Wanna do it?
I'm yellow tip.
Yeah,
Take it to the bottom.
I think it will be
There is
almost
very
all of
small,
this volume.
Okay, take it to the bottom.
Just put the tip to the bottom and I think this is 15 or 16.
हो गया.
Good. Put it all the way down. Down as much as
That's what I'm
करो balls पे डाल लो. Then it
नहीं
is
है, ये उसका वो है, illusion लग रहा है, हाँ नहीं है, कुछ भी नहीं है.
बस
I think it's good, right?
ये ऐसे. देखो हमारी ये वाली पाइपेट की टिप यूज करती है.
ये दूसरी इसके तो दूसरी
इसके
टिप्स
ऐसा
में
दूसरी टिप्स आती है.
This is also ready.
So we need to keep it in the
Uh,
ice or
I think we can do it now. Yeah.
यस यस.
Yeah, everything is
so
ready,
first
I guess.
Yeah.
Put the final blow.
Okay.
ये लो टिप. ये वाली ना.
Okay. Oh, do you want to do it? I think you did really well. Close and long. Yeah? Okay. Big honor. I mean, it can, yeah.
मैम ये लो. पहले वो एंटी क्लॉकवाइज करके वो कर सकते.
No need to be scared.
So first we need to add priming. First I have to 800 microliters. आधी फुल का है. कैसे ब्लैक बोल गया.
So did we have it like this last time? Or the other way around? So the pot is here, right? So let's just do it step by step. I say what to do so we don't...
Yes sir. You tell me what to do.
जी दूसरे बनाना.
फोटो ले ली.
Insert on the clip we did this. Okay, you were just sliding this port open, right?
Yes.
So now we try and suck out 20 to 30 microliters to make sure that, yeah.
मैम टिप चेंज
So
कर
you see
लेना.
where the port
फोन को
is,
टिप
right?
होगी.
इसमें कंटामिनेशन की कोई बात नहीं है, बस वो निकालना है.
So the thing is, you can hold it like this if you want,
Yes.
right? The thing is only turn, don't press.
Yes.
Yeah?
दे दे दे.
Till we can
कर
see
ले.
a little bit of, yeah. And anti-clock, yeah. Yeah, stop. Okay, see there's a little bit liquid, we pull it out. Okay?
Then we will be discarding the tip.
डिस्कार्ड ना.
Yes ma'am.
Then sir have to set it for 800.
Wait. Just make sure we do everything, right?
Yes, sir.
So we did this, okay, take
Load
800 microliter. So that's the priming mix.
टिप 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 मैम वही दे दो प्राइमिंग मिक्स
मैं बना रही हूँ मैं
बना रही है
That's great. Huh? So we try and add this into the yeah, and just move down. Yeah, keep moving. Then you put stop and pull it away. Yeah, perfect. Now we wait 5 minutes. Yeah?
5 minute timer time 5 minutes timer
5 minutes are over.
Okay.
5 minutes are over.
Should we just go ahead with the photo?
yeah gently lift this spot on ma'am
Priming mix.
हाँ हाँ
This
sure
one. So 200. Okay.
put in the spot on the
Press it onto the port, the tip. Yeah. It's on the port. Yeah. Oh.
sorry sir
Yeah. You can slowly move down.
हाँ वो देखो जा रहा है
Stop.
now 75 spot on का खोलना उसका नीचे डालना प्राइमरी से डालना
Okay. So we need to mix the library first. So you go up and down a little bit prior to loading. Then we add it to the spot on port, just drop by drop. Ready? Okay, take 75. Okay. Then one drop.
Yeah, it
good.
is going
So
up.
ready in. Drop.
yeah
Yep. Yep. So. Yep. Yep. One drop. Yep. Perfect. Yep. See, this goes much better
yeah
with the old
then
dose.
that from there it was oozing out
Perfect. I think that's okay. Very good. Clockwise. Yeah.
gently replace the spot on sample port
Yeah. So we
Cover.
put it. See, as this pin.
Gently close the priming.
Yeah.
बस लाइब्रेरी को थोड़ा सा जो लाइब्रेरी का ट्रेनिंग
Then where is this light shield?
Light shield.
Here?
No, no, एक shield type.
Yeah, that I have kept in this.
Inside.
Yes.
थोड़ा ऐसा हो गया बनाना पड़ा था
सर वो तो मिल गई लाइट शीट तो मिल गई
These also.
Hardik, you are having pizza?
क्या है अंदर लाइट शील्ड
उधर से ब्रॉड
Making
Here?
Can we use something like this?
So, what if you can make it?
yeah, tweezers is the best. Shield it a little bit like this. It's at the very bottom.
वो ऐसे चिपक गया उसके साथ
Yeah, tweezers.
या वो आपके हाथ में पकड़ में नहीं आएगा उसका फ्लैट सरफेस है इसका कट दिस साइड चिपक गया
Let me.
Just cut it off.
Can you
मैं करूँ
ना अंदर घुस गया ना ये कोई लंबा चीज है This is
I think that's it. Okay, we are ready to
to go.
sequence. We can do this later.
चलो
ये
try
बात
करो
कर
इसको
लेते हैं
ये अंदर ही घुसा जा रहा है
This is like the last step. I think it's ready. Yeah. Keep it horizontal.
वीडियोग्राफर किधर राइट सो इट हैज डिटेक्टेड आई थिंक यहाँ सैंपल आईडी सो
So wait, let me have a look.
So experiment name is, I'm just saying here, and sample ID is 24.
Yeah. What's this is sample ID.
I can get the date 21st.
Yeah.
Yes, you are putting this one right.
Yeah. So it's DNA.
Okay, this is just short distance.
And walk back or back. Multiplexing, yes. And it just allows you to select the output. Just narrows the number of options.
So there is these two models. So which one?
Yeah, that one actually. Wait, actually, let's just double check. So on the box, where's the time? Yeah, that's. So SQK 16S 114.24.
ऐसा वही है
SQK 16S 024.
Yeah.
This one.
Yes.
1424 sir.
So let us just check.
No, it's definitely multiplexing, yes.
Okay. Then what about PCR free?
No, it's
PCR.
it's PCR.
So I think then this is the only one left.
So 1 to 4, yeah, PCI 1 to 4, that's the thing.
Yeah, that's it.
1 dash 424. 2 is not written.
1 to 4. B1 to 4.
I think it should be already this one.
Yeah, B1
This is
to
1 to 4.
So we normally use fast place.
Do we want alignment done right now?
No, I normally go with the default options. Okay.
So there is fast.
So we should have live fast.
Acha,
place.
fast base calling is here, right?
Yeah, right. Yeah, choose fast, otherwise won't work. Barcoding.
Trim off barcoding. Trim
Yeah,
off.
I think so.
Barcode both ends off.
I think it's both ends actually.
So what should I do?
I think barcoding is both ends. Can somebody look at ChatGPT and then that's.
गूगल में बस स्टैटिक पे लिखना है सब आ जाएगा
ये खुलता ही नहीं
सब चलता है
है। IT doesn't open or what?
Life is becoming ChatGPT right now. Everything just any question. I just type any question.
So what should
in
we
life.
check?
Just.
सब वीडियो कॉल कर लेते हैं इनको सर इसको वीडियो कॉल या आप डायरेक्टली कीजिए
So let me just send him.
व्हाट हैपेंड नथिंग हैपेंड नथिंग सेम
Same like
अच्छा यहाँ पे so min 114 दिखा रहा है वही लेना है ना min 114 HD नहीं लेना ना
इट्स ऑल ऑन
ये क्या लेना सैंपल आईडी
DNA,
अच्छा
PCR, multiplexing.
जस्ट वेट
in this video as well. We can do as per this also.
YouTube. There is DM right now.
No, this Oxford monopore.
हाँ फ्लो सेल min 114 ही
Call
लिखा
him
है
directly and he could just guide us through.
sorry just one second so DNA में select करना है ना DNA PCR and हाँ now we
Yes,
are
sir,
getting the right thing
multiplexing भी हमें okay
yeah,
करना
now we are getting the right thing
Oh yes, so now we got it.
हाँ 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 अच्छा ये बताइए कि अब इसमें सब चीज़ बस चालू कर देना बारकोडिंग बोथ एंड्स ऑफ है ठीक है हाँ नहीं अभी वो इट इज़ टेकिंग द राइट किट नाउ इट इज़ राइट ठीक है ओके 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 थैंक यू हाँ मेरे को आया था मेल भरूंगा मैं है ना भरूंगा ठीक है ओके थैंक यू वेरी मच So I think we can just start this. There's a call कर रहा है.
अब यहाँ से हमने उस टाइम वीडियो लेकर हाँ
So now it will happen. So we don't need to.
Hopefully, yeah.
So I just restarted this Minnow and it selected the right kit then.
Oh, really?
Yeah.
That's it. Okay. Then can you give them some electrolyte water?
Go to.
ये वो S12 है उसमें S18 उसमें 12 लिखा होगा वो ध्यान रखना Can you pass me the vial that is containing more
This is your water, maybe write W on the top. Yeah, then you can each do one, yeah? So S10, we want 34.7 microliters of 34, so
Point seven should be
34, then 0.7. So that's
नहीं उसमें 34 में और 0.7 दे रही है
आप आपसे 34.0 करो
and that's in the sample pen.
तो क्या देखो
Yeah. So in here, yeah.
Sir, 34.7 we need to take the water.
Oh, wait, sorry. No, sorry. Actually, good point.
Yeah.
No, so water you need 5.3.
Okay. 5.3.
DNA.
Very small volume.
Yeah.
So next one is close the lid.
पानी भी पीना.
The next one is 12.2.
So we need to change the tip or we can use the same?
Because it's all water, you can use the same. So maybe someone else can try.
Yeah. For sample 11.
Yes. The next one is 10.2. Sample 12.
10.2.
10.2. Next one is 22.8.
I will be needing the green, the orange one. 22.8. Tau is it? So we will be taking 22 from here. The 22.8.
Yeah.
So we will be taking 22 from here and 0.8 from here. Tip ले लो. So next.
31.9.
So this is for S17.
That's 17, yeah.
How much?
31.9.
31.9. 30 or 41? 31.
31.9.
31. 0.9 यहाँ से ले लो. अब 0.9 डाल दो. आगे आगे done.
And then again 31.9% lately. Same.
31.9.
Then 7.8. And one of the girls.
Yeah. 7.8. cards with different calories.
We need to wait a little bit. We have the values for the qPCR.
I go hearts and
Yeah, we need these two.
so
It's like PCR again, we are copying the gene, yeah? But there is a dye that attaches to the DNA. And if only if the dye attaches to the DNA does it produce a signal. So the more DNA we have, the more genes we have, the more signal.
yeah.
Yeah?
जितनी fluorescence लगेगी तो उतना quantification होगी.
So what we show here is how many PCR cycles. So from 0, 5, 10, 15, 20, 25, 30. Each cycle we should double the number of genes.
Yes.
Yeah? So if we have
Like DNA replication.
If we have 1, we have 2, 4, 8, 16, 32, 64, and so forth. Yeah? Okay. So if I have only one gene in my vial, it will take many, many cycles until I can see it. Yeah? So in this example, the lowest standard, I think is standard 1. So this standard. So I shouldn't have done that. Anyway, so this standard with very low concentration, it will only show up after many, many cycles per minute. So this is what we call CQ value. Yeah. The CQ value is when the signal crosses this line here. So when you start seeing the signal. Whereas this standard here has very high concentration. Standard 8 is the highest concentration. So we see after 9 cycles. And then you can translate this CQ value into the concentration, because for the standard we know what's the concentration. So this standard here, standard 8, has a high concentration, 10 to the 8 genes, and it showed up, I showed you before, CQ value 9.2.
9.2. Yes, yes.
Right? And then this is 10 times diluted, so this is the concentration, and this is the CQ value, and so forth. So this is not the best calibration curve, but it's kind of showing you how it works. And then once we have this calibration, we can then have the CQ value of our sample. So now we know the CQ value and we can determine the concentration.
Yes.
So that's how qPCR works. Let's see if we have a better calibration curve. This is very good calibration curve. Yeah? So you see very linear, the two standards agree. Then, so these samples, do you think they have high or low concentration? So CQ value is 30.
Yeah, and they have low concentration.
So that's
These are high concentrations.
okay, let's see. Oh, great. Are you sure these are the ones that we put in the machine? Oh, great. So So because these we are already familiar with.
Yeah.
These are like a more like communicate that machine.
Okay. So can we have more? Because we have 48 samples. Now we have even more. We have 50.
अरे मैंने पता नहीं था, मैंने चेक नहीं करा था उस वक्त.
So each is 8.
It is 48. No, it is 48.
Yeah, so can we have like double this? Can we have more?
So we can also compare them with the earlier used.
These first.
It's 10.
So let's leave this one out.
But we are clearing the dilution. So DNA is defrosting and
So sample 11 we need 34.5.
the first one was S10.
Sample 10.
Yeah.
34.7 microliters.
34.75.
34.7.
You can pick 34 and 7.5 from here.
No, just 7.
0.7.
0.7.
0.7.
It just diluted to 5 nanograms.
7 milligrams. Sir, it is 34.75.
7.
Okay. 0.7. Note it. So this is for S10.
S10.
So is it 0.75 or 0.7?
0.7.
Because it has double.
Defrost for the plus.
Okay, to the computer, really nice.
Changing the display of
I hope it's only the display. If this went to sleep, it's not a good news.
Yes, sir.
Finish.
Start, sir. Next.
Half to need double time.
So we make 40 microliters. Oh,
So next,
I-40. Okay,
I think
I-40.
it's a complicated calculation. So she divides the concentration by time. That gives you how much you need to dilute it. Then they make up a total of 40. 11 is 27.8. I didn't check, I just crossed it.
ma'am, यहाँ से 27.5
You can do it easier.
बाकी 0.8 जैसे आप point is 40 but take it coming here. No difference with that, so you get how much?
So this is concentration divided by 5. So you need to dilute 1.5. So we don't take 40, we take this much less.
40 divided by this 1.152.
Then this is 40 minus this.
Bottom पे जाके
So much work. We don't lose them. missing data. Do you have one of your own samples or
Yes, sir.
you added one? Which is your one at the end?
S17.
He's not there, I don't know.
So next S12.
S12, right?
Yes, sir.
29.8. This is 6.7.
Number 17, I think ours. Number 17, I think our sample.
No, it's number 12.
Sorry.
So yours will need much more dilution because your concentration is high.
Five जाने वो ले लो
By yourself, maybe you need only the and contrast time if you know what you do.
Yes, sir. S13.
7.
Ma'am, 17.2. मेघाली में मातो क्यों आपका ही sample?
17 is 8.1.
8.1.
Yeah. 8.1. It's your. We need to dilute the other.
8 and 0.1
So somebody who's very good at pipetting can do the standards, and the others, everybody can help with some of them. So 12, I think this. So maybe use this one for 12. And you don't need to change the tip.
So which one is a master fix? This one?
Yes, ma'am.
Can you show me just to check? Yeah, I think that's it. Yeah. So guys, I can spin it down first if you want.
मैम बस इसकी ना एक चीज़ याद रखना ये जैसे हम 6 wells empty
Can you label all of it?
yeah
Yes, so we are fine.
S24AB we have made replicas also
स्टैंडर्ड किसमें डालेंगे
standard 3
डाल दी
डाल के
तो नहीं चाहिए
नहीं
Did you do the first one?
yes sir
Yeah, okay. And we can check.
this closed it
Yeah.
मैम देखिए बोल रहे चेक एक बार ऊपर उठा के देखो ऐसे नहीं मैम वो
No, just lift it out. Lift out the tube. lift the tube out if you want to see which one is liquid.
साइन करो
This one here. Okay, so you can each maybe do, I don't know, 4.
सबको चार
Yeah, just keep feeling the qPCR mix first.
We need to sell this also. Yes, ma'am.
All the wires need
बस
to
DNA
pass.
नहीं ऐड करेंगे NTC में.
सब लोगों को चार यहाँ से शुरू याद रखना जो भी लास्ट में लगाएगा यहाँ से ऐसे NTC A से
मैं इधर से देखती हूँ आपको.
In theory, we should do this in the
Left arm. Last 4.
Okay, that's one. That's fine. Then do another, the second one.
No,
No,
same.
because it's the same. Yeah,
It's second one. Now we need to change the pitch because we will use standard pitch.
stand up 2. So we do duplicates, right? So it goes in 3 and 4.
टिप है
चेंज कर लिए.
लैपटॉप कितना गंदा हो रहा है लैपटॉप के कितना गंदा हो रहा है
मेरे भी है लैपटॉप
Now standard 3.
बड़े जो है ना इतने बड़े जो
ये जाएगा 5 and 6
Yes.
हैश में